Hello everyone, and welcome to the video. Today we are talking about this topic of what we call the central dogma of biology. So this is kind of an intro to this idea of what we consider to be the central dogma of biology, and we're also going to talk a little bit more specifically about RNA today, which I'll get into in just a second. So you may have heard the word dogma used before if you've ever maybe heard it in relation to religion or theology in some way. The definition of the word dogma is going to be an important belief or an idea held by a certain group. And when we think about the central dogma of biology, scientists kind of coined this term when we thought about some really important ideas related to biology, some really important processes that occur that allow for life as we know it. Again, remember that biology being the study of life, this is going to be a really important principle for this study of life. And when we sum it up, we break it into three parts. So the central dogma of biology is when we think about the process of information in DNA being transferred to RNA and then being used to make protein. So this is kind of our three-part process here, thinking about how all of these different things can be used to create life. And again, the reason we call this the central dogma of biology is this process describes how the genetic code in our DNA is used to create proteins, which are the foundations of living things. So if we remember earlier back towards the beginning of the year, we talked about how proteins are one of the four major macromolecules that we find in living things. And proteins go into building the parts of our body. There are enzymes for our cells. Proteins fulfill a lot of really important functions in living things. So when scientists figured out how this process works, how we go from DNA to proteins, we kind of coined this to be the central dogma of biology. So it's a really important topic and one that we're going to spend a bit of time on explaining how this process happens. And there are two main processes that we kind of break down the central dogma into. The first one is going to be what we call DNA transcription. When we transcribe something, or this process of transcription is basically that means copying something. We are going to be making a copy. And in this case, we are taking the genetic code of DNA and we are copying it into a molecule of RNA. And RNA may be a new molecule to us. I'm going to dive into the specifics of RNA in just a second, but we see this first step of the central dogma here. So we have our usual double-stranded DNA, and we're going to use the process of DNA transcription to go from DNA to RNA. So we're going to end up with this molecule called RNA here. And then the second part of our central dogma is going to be what we call DNA translation. And what we're going to do, we're going to use this RNA that we just made in DNA transcription, and we're going to use it as the code to make proteins. So we took the genetic code that was in DNA, we copied it onto this molecule called RNA, and then we're going to use this molecule of RNA to serve as our code to make this molecule of protein here. So the information contained on our molecule of RNA is going to serve as a template or the code when we create this protein down here. And again, going from DNA to RNA to protein is what we call the central dogma of biology. To quickly point it out as well, we remember our previous topic that we just talked about is DNA replication. That's kind of shown here, showing that we start with DNA and then we use DNA polymerase to go back and make another molecule of DNA. So this is a related process that we kind of talk about as the same time as these other two processes. But just keep in mind, when we do DNA replication, we have DNA and then we end with DNA. So just an important connection there. So I mentioned here RNA, and I want to take a little bit more time to talk about RNA and thinking about why it's important, how is it similar or different to things that we've already talked about so far. So RNA stands for ribonucleic acid. And hopefully this sounds very similar to you to another molecule that we've talked about. We've talked about DNA, which is going to be called deoxyribonucleic acid. So even their two names are very similar. So when we think about the structure of these two molecules as well, it is going to be very similar. DNA and RNA, they're two very similar molecules. I like to think of them as kind of being cousins in some ways because there are a few key differences between these two molecules. First off, there's a different type of sugar group in these molecules. And our sugar groups, if we look more specifically here, is going to be our pentagon-shaped group here. Remember that we have nucleotides in our nucleic acids. RNA is nucleic acid. It's going to also be made up of nucleotides, and nucleotides are made up of phosphate group, a sugar group, and a base. So when we have this sugar group down here, the sugar group is slightly different. In RNA, we call it a ribose sugar. But in DNA, we call it a deoxyribose sugar. And this sugar group is more or less the defining factor of why we call RNA RNA and DNA DNA. It's based on the type of sugar group that it has. So RNA has this ribose sugar, that's where the R comes from. And DNA has deoxyribose sugar, which is where the D in DNA comes from. Another major difference is that RNA is single-stranded. So this is an example molecule of RNA, and you'll notice that there's only one strand of it here, and this is its natural state. We're not going to be replicating a secondary strand to basically match up with these base pairs here. In its natural state, RNA is single-stranded, and on the other hand, DNA is double-stranded. So it's one of the main differences between them. RNA is single-stranded and DNA is double-stranded. And then the last main difference we're going to point out is that RNA does not have thymine, T. Instead, we are going to see that RNA has uracil, which is going to be represented with a U, and uracil is going to bind with adenine, A. So in DNA, the four major base pairs that we find are going to be cytosine, guanine, adenine, and then we usually find thymine, T, in DNA. However, RNA is different. We see here in our example picture, we're going to have a U instead. So we also see the U's popping up here. And I like this picture because it actually shows this single molecule of RNA binding to itself, which actually does happen. And we see here that A matches up with U. A matches up with U. So when we think about the differences between RNA and DNA, the thymine that we usually find in DNA is going to be replaced with this U, this uracil here. So that is the last main difference between RNA and DNA. Otherwise, they are very similar. Again, they're both made up of these nucleotides. They're both going to have this sugar phosphate backbone, and they're going to have these nitrogenous bases in the middle. So they are similar, but we do want to keep in mind these few differences here. And then the last thing I want to talk about in this video is that there are actually three different types of RNA that we're going to be focusing on going forward. So the first main type that we want to keep in mind is this molecule called mRNA. And this M, we're always, whenever we're talking about different types of RNA, we're going to put a lowercase letter in front of it to differentiate between the different types. This lowercase m stands for messenger RNA. And if you know what a messenger is, it's someone or something that carries a message from one thing to another. And that's exactly what mRNA does. So messenger RNA is a copy of DNA created in DNA transcription. And it's used as instructions to make proteins. So this is a picture of our mRNA down here. And it's going to look very, very similar to basically a single strand of DNA. Again, remember from that previous slide we just talked about, there are those few major differences between DNA and RNA, but mRNA is going to basically carry the information that was stored on DNA, and it's a copy of that DNA genetic code. So mRNA is our messenger RNA, and we use it we create it rather during DNA transcription. Our second main type of RNA is called tRNA. And this T stands for transfer RNA. So when we have transfer RNA, this is going to bring amino acids to a ribosome during DNA translation in order to create a protein chain. And we're going to talk about this when we talk more specifically about this process of DNA translation, but this is a picture of our transferred RNA, our tRNA. And you can see here that these bases look very similar to the bases that are a part of this mRNA here, but they're also holding this group up top called an amino acid. And if we again think back to our macromolecules unit earlier in the year, amino acids are the monomers of proteins. So amino acids are the building blocks of proteins. You put a lot of amino acids together to make a polypeptide chain, a long kind of chain of proteins. that is our second type of RNA. And then our last one is called rRNA. I know it's a little confusing to have two R's, one right after the other, but this R here stands for ribosomal RNA. So this is going to be RNA that combines with proteins to form ribosomes. So we see it pictured in here. The rRNA is going to be these strands inside of our ribosome structure, and this basically allows ribosomes to have their function. So the rRNA combines with the protein. This blue and yellow part is representative of protein. So they combine together to form what we know as ribosomes. And these together, as we hopefully remember from our cell parts unit, ribosomes are responsible for making proteins. So this is going to be really closely tied to our process of DNA translation, which again we'll talk about in a different video. So I know that it may be tough to consider these three types of RNA kind of outside of their context, but I do think it is good to kind of get introduced to them now, and then I'll be bringing them up in later videos. Awesome. So that's all that I have for now. Thank you for watching.